So, good morning and welcome everyone to this morning, to today's morning session. My name is Edvinas Orentas, and I will be chairing this plenary talk. And I'm very happy that everyone feels like learning about the latest advancement in the field of single molecule microscopy is an ideal way to spend a Friday morning. So it is a joy introducing to you Dr. Viktorija Glembockytė, who is here from Munich. And I think we first met like more than 15 years ago, exactly, when I was a mentor of Lithuanian Chemistry Olympiad team for high school students, and Viktorija was a team member competing in national Olympiads and abroad, and I could tell that she had bright future ahead of her even back then, and it proved to be accurate, quite literally, because she worked with so many bright molecules during her Ph.D. studies with Professor Cosa at McGill University in Canada from 2011 to 2017, so attempting to decipher the molecular mechanism behind the photobleaching phenomenon, and then to devise effective methods to prolong the lifetime of organic chromophores, which is a key parameter for successful application of a single molecule fluorescence. And we witnessed an instance of this knowledge applied in yesterday's talks of your student. So then, following a brief postdoctoral fellowship at McGill University, she joined a group of Professor Tunable at Ludwig-Maximilians-Universität München in 2018 for a longer stay, and here she was introduced to the powerful concept of DNA origami and began to apply this method for creating intriguing nanoconstructs for a variety of uses, and often in conjunction with single molecule fluorescence. And she now holds the position of junior research group leader in the same group since 2022, and as far as I know, she will be establishing her own independent research group this autumn at Max Planck Institute, Heidelberg. So her current research focuses on molecular system with interesting application at the interface of chemistry, photophysics, and biology. And today she will be talking about nanoscale tools for sensing and imaging single molecules. So please, let's welcome Viktorija on stage.
Labas rytas, good morning everyone. Thank you so much for this really kind introduction, and thank you so much for inviting me here. It feels really special to come back to Lithuania to share with you my scientific adventures in the last few years in Munich. And also it feels really special to give a talk in Lithuania because it's my first conference. So it's been a fantastic week, and congratulations to all the organizers. And today I will tell you a little bit about our efforts to build different tools to image but also sense molecules on a nanoscale. And one of my favorite methods to do that, and I'm very biased because I got hooked on this method in my PhD studies, is single molecule fluorescence. So for those who are not so familiar with single molecule fluorescence, what we can do there, we can label our biomolecules or even materials with very bright fluorescent labels, and then we can do all sorts of different applications. We can track motor proteins, for example. We can also use different donor and acceptor fluorophores and then study rotational and conformational studies using energy transfer. We can also study interactions between different biomolecules, let's say nucleic acids and proteins. We can even do a catalysis, study catalytic processes on a single molecule level and look at the processes that are unsynchronized. And just one of the highlights of really big applications would be real-time single molecule sequencing, which also stemmed from the field of single molecule fluorescence. So when I got into this field, it was all really exciting because this kind of method basically provides a window to the nanoscale world. But for me, it got even more exciting moving to Munich, because now instead of just observing a nanoscale world with the tools of DNA nanotechnology, we can also start being a participant in the nanoscale world and create different functions. And today my goal would be to show you a little bit of the synergy of this ultimate sensitivity that the single molecule fluorescence provides and this ultimate ability to place different functions on the nanoscale very precisely. So I will talk about a few different projects, but I wanted to start with this project that was my first project in Munich as a postdoctoral researcher, where we tried to build signal amplification tools to make single molecule fluorescence, to bring it closer to perhaps point of care applications. So why do we need really signal amplification there? So I told you about this fantastic applications of single molecule fluorescence imaging, but detecting single molecules is actually hard. And the way I like to illustrate this challenge is with this where is Waldo problem. So when we try to detect a single molecule, basically what we're doing, we're trying to see one molecule in Avogadro's number of other molecules. And you can imagine that the main problem is really signal to noise ratio. How do we see that one molecule? So luckily, scientists are very smart and they came up with a lot of different tricks over the years. So the first trick is to use really bright fluorescent labels. So we label our biomolecule we want to detect with a fluorophore. This fluorophore will emit millions of photons. We want these fluorophores to have high quantum yield, high extinction coefficient, and high photostability, something that was the topic of my PhD studies. And then this is not enough because we still have a huge volume of molecules. This one trick alone is not enough, so then we play a second trick, and the second trick is we reduce the observation volume. Instead of looking at really large pool of molecules, now we zoom in a little bit, and we can do that by using total internal reflection microscopy, where we excite our molecules immobilized on a glass surface, and only the molecules at the surface are excited. We can use confocal microscopy, where we excite the molecules with a focused laser beam, or we can even use nanophotonic devices. However, this second trick, what it results in is the setup like this. So basically, up until recently, most of the single molecule applications were really limited to high-tech optical laboratories. It required highly trained personnel, and it's not surprising that there were not that many applications of single molecule imaging in point of care. So when I joined Philipp Tinefeld group in Munich, the challenge that was posed to us was, okay, can we detect a single molecule on a smartphone camera? Could we see one fluorescent molecule with a phone? And this was really crazy, and I thought it will never work, and that's why I actually joined this project, because it was very challenging. And there we had to play a third trick. And the third trick that we played, we said, okay, we need even more photons from our single molecule. So let's create an antenna. Let's amplify the fluorescent signal even more. So how did we do it? We basically used the very specific properties of the plasmonic nanoparticles. So plasmonic nanoparticles, when they're excited with a wavelength of light that's bigger than their diameter, what that basically leads to is the oscillation of these surface plasmon electrons. And you create an enhanced electric field around the nanoparticle. And what happens now when we place a fluorescent molecule in this enhanced electric field, if we look at the typical Jablonski diagram, we excite the molecule and it emits photons. When we place this molecule now in the proximity of the nanoparticle where this plasmonic field is enhanced, basically two things happen. One thing is that we increase the excitation intensity. So basically our nanoparticle works as a magnifying glass. And on the other hand, we also increase the rate at which this molecule emits the photons. So we increase how fast we cycle between this ground and excited state. And this way we can improve the fluorescent signal tens and sometimes even hundreds of folds. This electric field is enhanced even more now if we couple two of the plasmonic nanoparticles. So this enhancement of the electric field could reach hundreds. However, the tricky thing is now, how do we place this one fluorescent molecule, and how do we place a diagnostic assay that later we would run on a smartphone in between of these two nanoparticles? And this is really where the field of DNA nanotechnology comes in and provides the tools to do that. So for those who are not familiar, the technique that we used to do this was DNA origami. So the way the DNA origami works is quite similar to paper origami, except your building material is DNA. So how we start, we have like a really, really long circular DNA sequence. It's around 8,000 base pairs. It's from a viral plasmid. And then we know the sequence of this DNA. So we can use computer-aided design to basically come up with some connections, because we know that the DNA will form double-stranded DNA very specifically. And then we use short pieces of complementary DNA to bridge different parts of this long scaffold. And this way, we're actually doing folding, similar to paper folding, and we're assembling different two-dimensional and three-dimensional nanostructures. So the cool thing is that these short pieces here, we can functionalize. We can put fluorophores, we can put biological modifications, we can attach different nanoparticles. So basically, this DNA origami works kind of like a molecular breadboard. You know where these functions will end up on a nanoscale, and you can put different functions and then start to see how they interplay. So with this approach, there's like number of really exciting two-dimensional and three-dimensional nanostructures that have been created, even like nanomotors on a nanoscale, or like you can even start assembling these DNA origami together. So this example, for example, build capsids to catch viruses. This was a very recent example. But for me as a chemist, this technique is really exciting because what it allows you to place functions on a nanoscale. You can put dyes, nanoparticles, proteins, functional groups. So what we did, we used this DNA origami to kind of build a scaffold onto which we will assemble our nanoparticles and fluorescent molecules. So it was our scaffold. So we built something like this tower-like structure, and then we can immobilize this tower-like structure on the surface via biotin-nitrovidin interaction. So we introduce biotin modifications here. Then we can attach plasmonic nanoparticles. We functionalize nanoparticles with complementary DNA strand. We introduce the tagging strand so we know where the plasmonic nanoparticles will bind. And now what happens when we put the dye, we place also via DNA modification, we place the dye on this plasmonic nanoantenna. We see this would be a typical signal of the fluorescent dye when it's just on the DNA origami without any plasmonic nanoparticles. Now if we put a single plasmonic nanoparticle, we can increase fluorescent signal tens of folds, and then if we place it in between two plasmonic nanoparticles, we can increase the fluorescent signal up to hundreds of folds. So that is exciting, but now we needed to somehow make this place where we put this fluorescent dye accessible. So these nanoantennas have been studied in the group for a really long time, and the main problem why they haven't been used for diagnostics was, if you look here, this basically region where the most of the enhancement happens between the nanoparticles was completely blocked by the DNA origami. So when I joined the team, what we did together, we basically reinvented the DNA origami nanostructure, and we made nanoantennas that had cleared plasmonic hotspots. So instead of using one tower, now we use this kind of two-tower-like structure that has a big gap in between. And then the nanoparticles would come and bind from the front and from the back, and then from the top, you would have access for different biomolecules and analytes to come and bind in this cleared space in between the nanoparticles. Here you can see the electron microscopy image of this, and you see the design structure and the structure that we got. You see like how well you can predict what kind of 2D and 3D structure one can design, and even the nanoantenna. This is the TM structure of the nanoantenna. So it's quite, for me, it was quite exciting to see how precise you can get on the nanoscale. But what was really exciting is that we still had a very high fluorescent enhancement. Despite the fact that we cleared this plasmonic hotspot, our average fluorescent enhancement was still 80 to 90 folds. So now we have a cleared region where we can try to put a diagnostic assay. So we designed this proof of concept diagnostic assay where we, in this cleared region, we incorporated sequences, DNA sequences, that are specific to antibiotic-resistant bacteria. So if this antibiotic-resistant bacterial DNA is present, it would bind here in a plasmonic hotspot, and then it will bind in a way that it will create a little overhang, and then we would bring dye-labeled DNA imager strands, and then DNA imager strand will end up exactly in the plasmonic hotspot where we would amplify its signal. So the way we studied this was with single molecule fluorescence to show that it works. So we labeled the DNA origami with a green fluorescent dye. And then now when we add the specific DNA sequence and the imager strand, you see very bright red fluorescent dyes. And in this case, we ran this also in buffer, but also in serum. So the nanoantennas were stable in the heat deactivated serum. But I think the nicest thing is now if you don't put any target DNA strand, you see almost no false positive signal. And the reason for that is that this plasmonic hotspot that's created between these two nanoparticles, it has a volume of zeptoliters. So chances that this imager strand will end up there non-specifically are very low. So we had a very low non-specific signal. Another nice thing is that if you look now at the intensity of all the amplified spots that we detected, almost everything that we detect is higher than the intensity of the single dye. That means that we basically now differentiate from the single molecule impurities. So doing single molecule diagnostics, one of the problems is that your impurities are also single molecules. So if you can differentiate from that, that gives you a diagnostic advantage. So now we have an antenna that we can amplify the signal. We have a diagnostic assay that we put in the nano antenna. So the next challenge was, can we see these molecules once we detect them on a smartphone camera? So we had to actually engineer a device that would allow us to use the smartphone. And here, actually, the key piece of this device was a laser from Lithuania, this matchbox mini laser that we incorporated. So this box is like maybe this size. We took it to the streets of Munich. It runs on the power bank. Cindy actually took it to another city for a collaboration. But basically what it has a laser. We have a 3D sample stage where you basically just push in a glass slide. And then the cool thing is that we don't have any fancy objective here. The only thing that we have is an objective lens that costs $8. So we don't have a high numerical aperture. We don't have like a fancy setup. And then here we just had the Huawei monochrome camera. And here you can see the picture of this device. And now I will show you the movie. And this movie was actually acquired with just a simple smartphone program. And then you will see, I hope I can activate it. I think it's a laser pointer. There should be a play button. But I think we have to deactivate the laser. Yeah, so what you see now, maybe not so exciting, but for me it was really exciting. So you see a lot of spots that are blinking, and eventually they're photo bleaching. So I spent 6 years in my PhD trying to make these spots not photo bleach. But here what we wanted to see them photo bleach. And basically if we now sit on these spots and extract the intensity time trajectories, we see a single step bleaching. And when you see a single step bleaching, this is the proof that you're detecting a single molecule. And you see the signal to noise ratio is also not so bad because we have this enhanced fluorescence. So this was extremely exciting because now we were actually the first ones to detect single molecules with such a simplified device that really didn't require crazy optics, and the setup was not so expensive. So that was really exciting. It opened a lot of possibilities for further applications, but it was also really exciting because we made a realization that, I don't know, for those who are familiar with the single molecule fluorescence, 25 years ago when this field started, the Nobel Prize winner W.E. Murner defined this single molecule as guacamole because it's 1 over avocados number. And what we realized that 25 liters, now we detected the guacamole with nachos. And it was actually a coincidence, but it was a funny coincidence. So one of the challenges, and this is actually the question that I was asked on the streets of Munich when I brought this device, was someone who's not a scientist and said, it's all good and great. You can see single molecules, but isn't it the challenge to catch the single molecules? And that was a key crucial question because where the single molecule diagnostics is really powerful is detecting only few targets that might be present in your diagnostic sample. So if you have a blood sample of 2 milliliters and you have 4 targets in there, it's not just to see them. You might have the technology to see them, but it's also really difficult to capture them. And this is where this project is now going in the group, and it's PhD student Ranuka who's just finishing this. So what she did, she tried to improve the capturing capability. So this nanoantenna first has to capture the target. So what she did, she basically nano-patterned a lot of these antennas on the surface. So it maximized the chances that something could be caught. And then also incorporated this into microfluidics device. And then now instead of using a phone camera, we use just a diode array, really not powerful excitation source, but also looking now at the really huge field of view. Because if you have 2 milliliters of sample and you only have 4 targets, you need a large field of view to see these targets. So here you can see almost a millimeter field of view, and here under 1 picomolar of DNA target, you see a lot of single molecules that you can count, and then possibly bring this technology to really low limits of detection where the single molecule advantage could really be powerful. So I'm almost at the end of this section, so I just wanted to briefly summarize what we did. So basically we invented nanoantennas that now have cleared plasmonic hotspots. So this was the design, basically the nachos. Cindy, who is here, actually increased this clearance of the plasmonic hotspot even more. So this is nachos 2.0, so that it can detect even larger biomolecules. We're able to place proteins in there. So we incorporated in these cleared plasmonic hotspot different ways to detect biomolecules, and we detected them on a smartphone camera. And if you are interested in learning a little bit more about it, We did this during COVID. We had a lot of time, so we wrote a little review article. So if this is interesting, I encourage you to read that. However, when we were working on this technology, and every time we wanted to detect a new target in this plasmonic hotspot, every time we wanted to detect something new, we had to basically redesign that assay over and over again and do tedious optimizations. And this kind of encouraged us to think maybe DNA origami can be also a powerful tool to help us make a bit more modular ways to build sensors. And this is actually the second project that was also my Marie Curie project that I will tell you about. And then the challenges that we're trying to tackle there, like a very fundamental challenges that one faces when thinking of building a biosensor. So if you think of a typical biosensor, it usually has two different units. It has a sensing unit, which could be a receptor, naturally occurring receptor, could be some aptamer, and then that sensing unit will have to bind the target molecule. So that would be an essential part, the sensing unit. And then that sensing unit has to communicate with a signal transduction unit, because that binding event has to be relayed in some sort of either optical or electrical signal. And one of the main challenges, at least when building optical sensors, is that these two units are extremely tightly linked. So let me show you what I mean. So for example, if we have a protein and we want to use this protein to build a biosensor, let's say we can label this protein with a donor and an acceptor dyes that undergo energy transfer. And upon the target binding, there's a conformational change that occurs, and then we read it out as FRET. Now, the problem here is that the amount of FRET that we will see will be extremely tightly linked with the conformational change that happens. And then we cannot really place these donor and acceptor dyes arbitrarily, because at some point we're going to affect the sensing unit. So that leads to tedious optimizations where we place these optical labels to get a high contrast and not sacrifice the sensing unit. The second challenge is that if we take a naturally occurring sensing unit, it will have a certain affinity. It will bind this target with this hill type of curve, and it will have a certain affinity, and you will be able to detect something at around the window of this affinity. But let's say if you want to detect something at the nanomolar range here, or millimolar range, you have no sensitivity. So you kind of limit it to the affinity of the sensing unit. So these are kind of two challenges. And then you can start basically engineering the sensing unit, but then that's again tedious optimization. So we wanted to kind of build a platform that would allow you to build sensors a bit more modularly without these tedious optimizations. So what we wanted to have a sensing element, and then to be able to take any naturally occurring sensing elements, or to basically access this pool of occurring sensing elements, then have an orthogonal ways to tune the response window of the sensor without changing the sensing element. Taking the sensing element, using it as it is, having a strategy to tune this response window, and then coupling this with a signal output element that always gives you a high contrast, always gives you a high FRET contrast. So, and that's where we actually said, okay, DNA origami is perfect for that, because it allows you to modularly couple a lot of different functions. So the way we did that, we took a DNA origami structure, kind of like a hinge-like structure that can undergo a huge conformational change. So in equilibrium, it's around 90 degrees angle, but it can basically open and close. And then we said we're going to use this conformational change to have now a high optical readout. And then here it was a model sensor, so we used reversible DNA-DNA closing interactions where we can detect the DNA target, but then the sensor can close reversibly, so we're basically working around the equilibrium. And then we wanted to put a donor and acceptor dye to have this high optical FRET readout. And then we were hoping that when the structure is open, we'll have no FRET. When it's closed, we'll have very high FRET. So we were very naive at the beginning and said, let's just put the donor and acceptor dyes on different arms of the hinge. So now the problem here is we draw these structures in Blender or in the other softwares as these rigid rods. And here you can see a simulation of such a structure. It's basically huge. There's a lot of electrostatic repulsion between these two sheets, and it moves a lot. So what happens when you put the dyes now on each arms of this structure? Well, you make a very expensive magnesium chloride sensor. Basically, now the distance between these two dyes becomes extremely sensitive to this electrostatic repulsion and this movement on a nanoscale. So you get very broad FRET distributions that are very sensitive to magnesium chloride concentration. So we knew this is not going to work. We had to play another trick. So the trick that we played was this. So we said, okay, we need these donor and acceptor dyes to be always in a fixed configuration. So we incorporated them with a really short 8 base pair DNA-DNA hybridizing interaction. So if you just had 8 base pair DNA hybridizing interaction in a solution, this will always, it will never stay stable. But because this interaction is placed on these hinge arms, whenever the hinge is closed, you have high effective concentration of these two strands, and they always stay closed. And what that leads to, it like basically position the dyes in the perfect configuration for very high Fred values. So you see very narrow Fred distribution, like almost quantitative Fred, both at high ionic strength and at low ionic strength. So we use this high effective concentration here. And that basically enabled us to make a sensor that's in the closed configuration has extremely high Fred values. And now when we add the DNA target, an open has almost no Fred. So we got this almost digital Fred readout with a single molecule sensitivity. So now we basically studied different ways how we could tune this dynamic window. So this is challenge number 1. Now we have a high optical readout. Now how we could tune the dynamic window. So here first what we did, I will just show you how we studied this. We did this on the single molecule level. So we mobilized this DNA origami hinges on the surface. We would add different concentration of targets. Because it's an equilibrium process, especially at low concentrations, we would equilibrate to make sure we're in equilibrium, and then we did single molecule confocal imaging at different concentrations. And this is how it would look like. At the beginning, with no target, everything's closed, and then you can start counting how many molecules open. Like you can really say, how many of my sensors opened? And then we did that, we would count open fraction, and then we would fit this over the concentration range to Hill equation. And with the Hill equation, we could extract basically our affinity, where the sensor opens, and we could also look at the slope, because the slope defines a Hill coefficient, so how cooperative this sensor is. In some cases, you want to be able to sense over many concentration ranges. In other cases, you want to be very sensitive to specific concentration range, so it's important to be able to tune the slope as well. So now we started brainstorming, okay, we want to keep the sensing element the same, we don't want to change it, we want to come up with ways to tune the dynamic window. And one thing that we could do that you couldn't do in the typical sensors, we could use multiple of these sensing elements. Because we have a huge nanostructure, we could incorporate multiple of these closing interactions. So instead of putting 2 closing interactions, we could put 4 and 6. And basically, the more closing interactions you put, the more stable you make this closed structure. So you shift your dynamic window to higher concentration ranges because it becomes harder to open it. And then you can see here, you also increase the cooperativity, because it becomes sharper, the response, because now you need to open all 6 of the interactions and they interact cooperatively. Another thing that we could do, we could leverage this electrostatic repulsion that's intrinsic in the sensor. So if you now reduce the ionic strength and would you reduce the shielding of this electrostatic interaction, you destabilize now this closed structure of the sensor. So what you do, you actually shift to lower concentration ranges. So the sensor will open at lower concentration ranges. So that's like an opposite arrow in this Hill and coefficient and an affinity curve. And now the final thing that we did, we said it would be really cool to mimic allosteric modulation that happens in nature. So in nature, there would be a kind of like an inhibitor or an activator that would bind on the receptor, and then it would make the binding event more or less likely. So what we did, we introduced an additional DNA-DNA binding interaction, and in this case, it's just 5 base pairs that stabilizes this nanostructure in a closed state. And then here you can see by introducing this tiny stabilization, you can shift the response window almost 2 orders of magnitude to, again, higher concentration ranges. And in this case, we never changed the sensing interaction. The sensing interaction was always the same. So we just engineered different tricks how you could adapt the dynamic window, keeping the sensing interaction the same. And here, one other thing that we showed, and I thought it was really interesting, is that you can now leverage this multivalency to also be a bit more specific. So if you close this sensor with multiple interactions, these multiple interactions, each one of them will be sensitive to the off target. So what I mean by that, we looked at how sensitive we are if we're trying to detect a nucleic acid. And we bring a nucleic acid that carries a single nucleotide base pair mismatch or 2 nucleotide base pair mismatch. Are we better differentiating it? Because now instead of having 1 closing interactions, this mismatch gets multiplied how many interactions we have. And if you look here, this we did with the 4 closing interactions on the sensor. The blue is the real target, and you see opening at the nanomolar concentrations. With 1 mismatch or 2 mismatches, we could, bump up the concentration to 10 micromolar and the sensor would still not open. So this is basically 4 orders of magnitude excess of the target nucleic acid, and the sensor still does not open. So this is like really, for me, was quite impressive selectivity. But those of you who work with nucleic acids would say, okay, you introduced a GC mismatch. It's a very strong mismatch. Could we detect something that's a bit weaker? So we then tried to see how far we could push this, and we started comparing like really similar mismatches. We introduced a AT mismatch to very, very similar AT mismatches. And here you can see that up to micromolar concentrations, we can still very clearly distinguish the right target. Only at 100 times higher concentrations of the missed target, we were not sensitive anymore. But even then, when you're not sensitive anymore, the trick that you can play, you can say, okay, let's introduce more interactions, because then we become more selective again. So for example, at these high concentrations where we're not sensitive, if we now introduce 8 interactions, we again become sensitive. And I think what's really crazy is that these 2 mismatches here, the energetic difference between binding this and this strand is just within the KBT. So you really can differentiate small thermodynamic differences in binding. So it was kind of like a nice side discovery that we did. Okay, so far I showed you that we can tune the dynamic window, and I showed you the high signal contrast. The only way this would be useful modularly if we can now take this high contrast and make new sensors without re-engineering, because that was the goal. We didn't want to re-engineer these sensors over and over again. So the key question was, can we now modularly exchange this? So far, I only showed you the detection of nucleic acids, which there are methods to detect nucleic acids. So we wanted to know, can we detect antibodies in this way? So what we did, we basically closed this sensor in a closed conformation, and we incorporated now as sensing elements for the antibody, these 2 antigen interactions here. And then these 2 antigens are positioned in a way that it would bind the antibody in a perfect orientation with the distance that is needed to bind an antibody. So now if there's an antibody present, we put this DNA opening strands and the sensor stays closed. So the sensor is kept in a closed conformation by the presence of the antibodies. On the other hand, if there is no antibody, the sensor will stay open because there's nothing keeping these antibodies closed. And then again, we could do this, and I think that the coolest thing is that you can see in these confocal scans, we really preserved this high Fred contrast. So we didn't have to go and now re-engineer the optical readout again. We took the optical readout that we developed, and then now we applied it to the detection of antibodies. And here you can see we did it in buffer, but we also did it in heat deactivated serum. Now we also adapted, besides antibodies, we also adapted this approach to detect enzymatic activity. So you can imagine that you can clamp this sensor with a specific DNA or peptide sequence, and in the presence of the enzyme that cleaves that sequence, you can also keep the high contrast. We also incorporated aptamers to detect biomarker proteins. So in this case, we incorporated the platelet-derived growth factor aptamer, and we could detect a platelet-derived growth factor at picomolar concentrations. But I think what's cool is that you can start making more complex sensors. So here we always keep the contrast, but you keep the contrast even if you make sensors that are multiplexed on a single sensor. So here, for example, we incorporated the antigens for 2 different antibodies and kind of build in a molecular OR gate. So here, if both of the antibodies are absent, the sensor would open, but if either of the antibodies is present, you see that the sensor stays closed. So you can start thinking of building a bit more complex sensors, but still preserving this high Fred readout. So just to summarize, like it was kind of like a project that led to a lot of different directions, but the key idea was there that we really wanted to just take and decouple sensing from signal output. And we did it with the DNA origami. There's a lot of really nice directions of people doing it with proteins and semisynthetic proteins. We showed that you can have a very high Fred readout. We also showed that there are strategies to tune this dynamic window without changing the sensing interaction. And then we also showed that you can harness this multivalency to show that the system can be cooperative, but also quite specific in some cases. And then we also showed that without re-engineering this optical readout, we can extend this sensor to a lot of different targets, actually, without having to do these tedious optimizations. And where we're thinking of going with this now is also we are working on protease sensors with this. Because when you're designing protease sensors, there are a lot of protease sensors, but they all work in a very similar manner. It's like you have basically a peptide sequence where you position donor and acceptor dyes, and upon protease cleavage to this peptide sequence, you would read out the like either the signal recovery or the FRET change. The problem there is that the proteases are very sensitive to this binding region. So you cannot put these dyes too close. You have to space them apart not to affect the sensitivity of the protease. But at the same time, then you compromise the FRET contrast. And this is also an application where this TDS design is very laborious work to design these sensors. So we were thinking, could we just use our approach to modularly exchange these peptides? Because we have a sensor, we can keep this readout that's always high, and then just modularly exchange different peptides. And then we can use multiplexing if we want. We can use the ability of these sensors to specifically immobilize them on the surface. And one sensor that we're working now with the collaborators in the hospital is Adams 13 activity sensor. So this protease is involved in blood clotting disorder. So this is something that's kind of proof of concept right now that we're working. But my big dream would be to actually make something that's kind of like a proteolytic fingerprinting platform where you could basically modularly design a lot of these peptide sequences, immobilize these sensors, and then from small amount of volume, basically read out how different proteases cleave all of these peptides. Because when it comes to diagnostic purposes, one protease activity is rarely a biomarker because all of these proteases work in cascades. And sometimes to really understand what's going on, you need to read out and fingerprint this proteolytic activity on the multiplex scale. And I think DNA origami could be a powerful approach there. So this is kind of where we're going with this. And finally, I wanted to also talk a bit about the challenges in making different nanoscale tools using DNA and using fluorescent labels. So this is kind of the directions that we're also exploring. So you can imagine if you use DNA as a building material and you use fluorescent labels as your readout, there are a lot of challenges that are associated. So one challenge is that the fluorescent labels bleach and they're not so photostable. And this is something that Cindy discussed yesterday. And we have a lot of different projects where we work in strategies to photostabilize these sensors and photostabilize these imaging technologies. And I won't talk about this today. I will talk about the other challenge. And the other challenge is that if your building material is DNA, DNA is something that gets degraded in complex chemical and biochemical environments. And when it comes to DNA origami, the main problem is degradation by nucleases. So if you take these DNA nanostructures and put them in serum, depending on the aspect ratio and what kind of nanostructure it is, within maximum half an hour, they get degraded. So this is like huge challenge. And then there's a lot of different ways people are addressing this. So one way is to really think very carefully about the design. There are certain design features that make them more stable. You can think of some chemical modifications. You can cross-link them. You can come up with some modifications at the ends of the DNA origami. You can do solution treatments where you intercalate different things and make them less resistant to nucleases. Or you can even protectively coat them with polymers, for example, polylysine, or you can even coat them with silica. So there's some strategies that are being developed, but what we were thinking the key challenges there is what happens? Is the function or addressability still preserved? Let's say if you design a dynamic nanostructure like the sensor that I showed you, and now we coat it with a polymer, is this nano device like still running, still turning around, or is it now becomes inactive? So how addressable and how well we preserve the function of this device is one question. And another question is, all of these strategies that I showed you, they kind of like slow down the damage, but they don't really reverse the damage. So we were interested already at the beginning of my postdoctoral research, can we basically come up with ways for these structures to self-repair? Like if they get damaged, can they self-repair? So instead of like just prolonging the lifetime, could we like also have some active repair? And then there's some results that I will show you here. And then the first one is this idea to cover the nanostructures with silica. And this is a collaboration that we had with Emily Hore-Jungmann group at Max Planck Institute for Biochemistry in Munich. So her group is working on this like really beautiful approaches to take DNA origami structures and then grow silica on them. So then you basically have addressability of the nanostructure, but when they grow silica on them, they improve mechanical, thermal, and chemical stability. These structures, you can throw them in serum and they're perfectly fine. But the key question was here, are they still addressable? So like what happens now if on this structure you incorporate little handles, like I don't know if you can see it here in yellow. Let's say we put 3 handles here. Can we still hybridize something to these handles? Can we still functionalize them with something? And this is where we teamed up with Emily and we did super resolution measurements DNA paint, basically what Cindy introduced yesterday, and maybe for those of you who weren't here, oh, sorry, I have to go back. Yeah. So for those of you who weren't there, like if we were now to just put a fluorescent label on the structure, it's only 180 nanometers. If we were just to put one fluorescent label, this would be a diffraction limited spot. So we had to do a super resolution measurements. And here we did DNA paint, and DNA paint basically has a single-stranded handles, and then you have imager handles in solution that bind and detach transiently. And as they bind and detach, you can localize them and pinpoint them, localize them and pinpoint them. So this is how you can get this, from this type of structure, a 3-spot image here. And the first thing we did, we took this kind of structure that had 3 single-stranded handles, and we did the DNA paint before silicification, and you see 3 spots. And then we silicified these structures, proved that they're silicified, and then we still saw 3 spots. And this was extremely exciting because that meant that now we have silica-coated structure with full addressability of all of these handles that we included. So we wanted to dig deeper to really understand what's going on. And here we designed a little bit different DNA origami. We designed a DNA origami that has only one long handle. And then this long handle can bind basically 3 different imager strands. There's one strand that can bind at the very top. So we said, okay, this will be accessible. It's probably going to stick out of silica. This imager A will be accessible. Imager B, we said, okay, we are not sure if we will be able to see the imager B. And we said, okay, imager C is at the very surface. We're probably not going to see it. So we're trying to understand how long this handle has to be for us to still be able to access it. And then when we did DNA paint, what we observed basically that the kinetics of binding to each one of these strands was exactly the same, whether it's silicified or not. So what that basically meant that each one of these strands was fully accessible. And the reason there is because when the silica grow happens, the grow happens primarily like, first what happens, the DNA origami gets silicified, then double-stranded DNA gets silicified, and single-stranded DNA gets silicified last. And if you control the kinetics, you can actually specifically silicify DNA origami, but not silicify the single-stranded handles. And just to show you that this is really not only some of the events that we observed with the same kinetics, we also labeled this DNA origami with a green label, and we looked how many of these spots had a red label, like what was the colocalization. And you can see here that colocalization was almost exactly the same whether we silicify or not. So that basically was really exciting because it said, now we have a robustness of the silica material, but addressability of the DNA origami, and we still have these fully accessible sites that we can functionalize. So now we're working on like trying to apply these silicification strategies to our sensors to prolong their stability in serum. And then the last thing that I wanted to talk to you about is these strategies to maybe reverse the damage. So here, in nature, this kind of self-repair and self-healing and self-regeneration is very common. Like basically a lot of systems in our body constantly self-repair. The photosystem too, and a lot of other examples. And we're wondering, why can we do that also with DNA nanostructures? Because the way these DNA nanostructures are built, they self-assemble. You put all the pieces together, you heat it up, and then they self-assemble. So if there is the self-assembly nature, we're wondering, could we also use the self-assembly nature to make them self-healing and self-regenerating? So here we kind of identify 2 different strategies. One of them would be self-regenerating, meaning that there's constant exchange of building units over time. So we have some function, and that function is constantly exchanging. So here as an example, we did this DNA origami nanoruler. So basically it's a DNA origami structure that we use as a brightness standard. And it has 100 sites, so it's a very, very bright. It would have 100 fluorophores. If you just attach these fluorophores permanently, it will photobleach and it would be damaged. But what we tried to do, so you see this is initial, this is bleached. But what we tried to do, we tried to put them basically in a regenerative manner. We put them with a transient binding that they're constantly exchanging. And as they're constantly exchanging, they can be fully recovered. So now if we take this DNA origami and we bleach it, it recovers fully. So now there's like a self-regenerating mechanism where you can have self-regenerating approach. The other strategy that we wanted to show is that it is possible to exchange only damaged building blocks, because sometimes you cannot constantly exchange your building blocks. You want to like repair the structure only where the damage is happening. And then here what we looked at, we hypothesized if in DNA origami there's a strand break, and we introduce a staple that can fix this strand break via toehold-mediated strand displacement, that this should be possible, because that's how these structures assemble. So what we did, we basically designed a DNA origami structure that had these two spots in there, like 1 and 2. You see at the beginning there's 1 and 2. And then the third spot, we basically varied the damage that is there. We kind of left some nucleotides unpaired, and then we increased how big the damage was there, and see if we could bring now from the solution the staple strands that carry this docking site that would introduce a third spot. So if there's a damage, could we introduce the non-damaged third spot? And then here you can see that as you increase the damage in this third site, if you leave more and more of these DNA origami things unstapled, the new strand can come in and replace it. So that means if there is some mismatched stapling, if you provide the fully matched staple, it can exchange and repair itself. So the way we use that, we actually show that we can have a mechanically damaged long DNA origami rods, and if we incubate them with the missing staples, they basically kind of straighten themselves. Or here what we did, we took these DNA origami structures and then we put them in serum, and then we put them in serum basically without these additional staples, or we added these additional staples and let them repair themselves. And we showed that even after 11 days in serum, they were still kind of repairing themselves, and we could prolong the longevity of these nanostructures. So it was not complete, the self-healing mechanism, but we could demonstrate that this is actually possible in DNA origami nanostructures. And with that, I'm actually at the end, and I really hope that I could convince you that these tools are very complementary, and it's really fun to combine single molecule imaging and DNA nanotechnology, because on one hand, you have this ability to see molecules with ultimate sensitivity, and on the other hand, you have this ability to place molecules on the nanoscale. And then if you find this exciting, and especially if you're at the stage where you're wondering what to do in your scientific journey, as Edwin has mentioned, I will be starting my own group at Max Planck Institute for Medical Research at the end of the year. Or if you liked any of these techniques and you would like to collaborate, really just reach out. I would love to establish more connections with scientists in Lithuania. And that brings me to the last and most important slide, to acknowledge all the people who were involved in these projects. So I was extremely lucky to work in Munich in Tunable group that was very supportive, and most of these projects that I showed was really like a teamwork-based projects, and that's why it was so much fun to work on them. So in this DNA nanoantennas, we were leading this project with another postdoc, Katerina, and then Florian Steiner, who helped a lot in building the setup. Leonard designed the DNA origami structure, and we had a huge team of people who worked on all the directions in parallel to make it possible to see molecules on the phone. And these modular sensors, it was really Leonard and Martina, two main PhD students working on it, and Thea did some selectivity measurements. And on the stability on a nanoscale, it was really Michi driving these projects. And all of the collaborators and all the funding sources who supported me over the years. And since I'm in Lithuania, I also wanted to say thanks to all the Lithuanian teachers, both in schools and Vilnius University, National Students Academy for, I think we have a very special environment in Lithuania where you can really get interested in science very early on. And I realized that how special that is, being abroad for so long. So for everyone who's young scientists, I would say, take the opportunity, like go to the labs and really use this chance that we can start science so early. And yeah, thanks to everyone who enables that. And thank you for your attention, and I would be happy to take some questions.
Yes, thank you, Viktorija. It's fascinating research indeed. So the floor is now open to questions.
So this is completely not my field, but I'm interested about that camera that you built to detect single molecules. Can I use my phone camera to detect that, or is it like sort of specifically built camera that you used for the detection?
I think you could use any camera at this point. So when we started this project, so the reason why we used the Huawei P20 was that the Huawei had a monochrome camera. And usually if you don't use a monochrome camera, all of your light is basically split out in different color pixels. But over the years, the cameras became more and more sensitive. So there are groups who use a little bit more optics, but they can even see green and red labels if they use the color camera. So for example, Andrei Klimchenko's group, they make like these really huge nanoparticles. So there they can really see in the green channel and the red channel, they can actually study FRET if you use a non-monochrome camera. My feeling is that if your fluorescent enhancement is really high, you could probably use any camera. The reason we chose the specific camera was really at that point, with the monochrome ability to kind of combine all the pixels, we were hoping that this would give us the best sensitivity. And if you saw, the signal to noise ratio was quite good. So I personally think you could probably do it with any camera. We didn't install anything special there. It was just a Huawei P20 phone.
Thank you. Any further questions?
Thought my catching skills were going to be tested. Thanks for sharing with us, Victoria. So I have a question for you. You were mentioning how you all would like to be able to array your sensing elements. And I'm curious if you've thought about combining it with technologies that are used for next-gen sequencing that also have these sort of arrayed DNAs or other molecules that maybe you could use to sort of get that orientation that you want without necessarily having to create a whole new technology.
Yeah, without doing any spotting or some kind of space.
Right.
Yeah, exactly. I think that's the neat thing about DNA origami, is that you can control which sequences you incorporate on the bottom. And with that, you could control to what it will hybridize. So I don't see the reason why this could not be combined. You're talking about DNA arrays, probably.
Yeah.
That's the idea. Like when I was talking about the patterning, that's what I was thinking of, using perhaps a DNA array to already have multiplex capabilities, and then having DNA, like complementary DNA sequences on the bottom of the DNA origami, and then directing them that way where they will hybridize.
Yeah, I think that would
I
be
think that's
great.
possible.
And then the other question I have for you, so you were showing us all about this sort of hinged structure, so I just sort of think it like this, and I thought it was really cool how you were showing us that you could do like the Boolean operations to know whether or not you have like two different things or whatever else, and I was wondering, could you set up maybe something almost like a double hinge or something like that so that you could actually determine whether things are happening in series? So if you had like, say, one event that might close and then only allow another, say, biomolecule or something like that to attach.
We were thinking a little bit about that, more maybe from the opening side.
Okay.
Because what I did so far was that I always use the same sensing element, because the idea was, let's not change the sensing element that's doing the dynamic window. But one thing that you can imagine is that you can use different closing interactions, and you can maybe detect longer targets where the first part of the target will have to bind here, the second part of the target will bind here, the third of... I think that would be possible. I would say one challenge that I didn't discuss of DNA origami, and it's really a challenge, or like something that you always have to keep in mind, is whenever you incorporate a function in the DNA origami, it's not quantitative. So it's not 100%. It's maybe 95%, but it's not 100%. So when you're making sensors and these like arrayed sensors, especially, you have to keep in mind that you'll have a small fraction of your nanostructures that might not work the way you want them to work. Because if I have like now 5 functions and I rely on all of them for my final output, if this has 90% incorporation efficiency and this has 90% and this has 90%,
propagating that.
it's propagating. So that's something that you always have to keep in mind. Like in our case, for example, one thing that you could think is the problem is that maybe the donor or acceptor dye is missing. Doing FRET is a nice thing, because whatever, if the donor is missing, we're not seeing FRET. If the acceptor is missing, we're also not seeing FRET. So if we're just reading out FRET change, we kind of like nicely get rid of that challenge, if that makes sense. But if once you start thinking of a more complex arrays, this is something that you really have to be aware of, that there will be a small fraction of these sensors that might not do the array that you imagine. But I don't see the reason why this would not be possible.
Cool, thank you very much.
Yeah, thank you for nice questions.
Any further questions from the audience?
Yeah, I too was afraid you're going to throw it to me, but thanks. Yeah, also totally not my field, but I was curious, in the time traces where you show the step changes, the sort of the system looked really fast, but did you actually look at the dynamics of how fast these sensors can operate, and whether this is at all important, or is it for you, is essentially these second scales is all you're interested in?
You were referring to the hinge sensor, yeah?
Yes, the hinge sensor.
Yeah, we looked into that, and like that's kind of like, it's a good question, but it's not easy to address it, because so what we did, one of the things that we did, we wanted to show that the sensor works at all of these concentration ranges. So obviously the opening kinetics is very sensitive to the concentration. So basically, if we work now in like hundreds of nanomolar, like if our sensor works at the hundreds of nanomolar, the kinetics will be within seconds, like tens of seconds. And we measure that, like if we put 100 nanomolar, we'll open within hundreds of seconds. But these sensors that we pushed to lower our dynamic range, so to say, if you only have picomolar concentration of the sensors on the surface, and your target is present at the picomolar concentration, it's not the sensor itself that's the limitation, it's the kinetics that becomes the limitation. And there, of course, you're much slower. And the reason why I showed this equilibration step, when we do this Hill coefficient, like a lot of the times how people establish these sensors, they always add different concentrations, wait 5 minutes and measure. So waiting 5 minutes at 100 picomolar and waiting 5 minutes at 10 micromolar, these two things will not be at equilibrium. So we always waited for hours to make sure that we are really measuring at equilibrium. So you're completely right. If you design a sensor to work at picomolar range, and your sensor concentration is also picomolar, you will be definitely limited with kinetics, which is why I showed this with nanoantenna. That was our limitation. So this nanoantenna sensing, if we just did it the typical way, single molecule way, 100 picomolar of nanoantennas, add the targets and the concentration, our dynamic range was picomolar. Ranuka, who did this patterning, microfluidics, all of these extra steps, she pushed it to hundreds of attomolar now. So you will for sure need some ways to speed up the kinetics at these concentration ranges.
Okay.
But if you're not kinetically limited, it would be tens of seconds, the opening of the sensor. The reclosing is the trickier part. Because then you have to overcome the electrostatic repulsion.
Okay,
Yeah.
great, thanks.
No more questions? If not, then I have perhaps one.
Yeah.
I was surprised this silification works that well.
Me too.
Any sort of condensation of this type.
Exactly.
I mean, it's usually not that well controlled, but
Yeah.
I believe it works through templation because your
Yes.
silicates,
Yeah.
the esters, it's with positive charge.
So the way this works is usually that you add a little bit of the positively charged silicates.
Alkoxysilane, I guess.
Exactly, precursor. You let this precursor template,
Yes.
and then you add the rest of the silane. And this is like how long you let the template and how long you keep the silane, so you can tune the conditions to make this not aggregate.
Yes.
So when the structures are immobilized on the surface, it's easier. It gets hard when you try to do the silification in solution because there the aggregation can
cross-link everything.
cross-link and so
But does that mean that the single-stranded DNA, it's sort of, you have also silification, just this reattaches somehow later, or is it completely intact during the process?
We think it's completely intact because it's completely accessible. Like it can bind basically. So what Emily's group showed, she made like a DNA origami crystals that have single-stranded handles. She silicified the monomer crystals and assembled a large crystal out of them, which kind of implies that you really can do full hybridization still. So what we hypothesize, but this is just a hypothesis, is that what's happening, so single-stranded DNA, it's kind of like a wobbly. Okay. So maybe it would silicify if you wait for hours, but at the beginning, maybe it doesn't get covered in the precursor, and it kind of creates a little pore. So one way to test it would be to basically see what kind of size of the molecular target could still bind to the single-stranded DNA. Because if you bring a giant protein, maybe this protein will not still be able to access it. So maybe kind of probe this, if there's a pore created around the single-stranded DNA. This is something that we're doing, yeah.
So with more handles, you can perhaps
create a larger pore.
Invent silicone origami from DNA origami, right?
Yeah,
the next step.
Maybe.
Inorganic origami.
Yeah,
yes.
so
would be
this
nice to have the same modularity of these molecules also for some hybrid materials.
Exactly. So that's what Emily is working on, like creating like a carriers and drug delivery vehicles, and but this was like a very recent study from last year, so we're still trying to understand fully the mechanism why the single-stranded DNA really remains intact.
Yes. Interesting.
Yeah.
Okay, so join me thanking Viktorija again for a nice presentation.